Hello everyone, my name is Sandra Sacristan. I'm a third year PhD student at Rey Juan Carlos University in Spain, and I'm going to talk about the use of assisted evolution as a new conservation tool to advance flowering onset in Lupinus angustifolius. The current global warming scenario makes necessary the consideration of new conservation strategies to mitigate the effects of climate change. The concept of assisted evolution refers to all those activities in which there's a human intervention on any of the evolutionary forces to help organisms to adapt to environmental conditions. Since flowering time adjustment is a crucial response of plants to global warming, the main objective of our study is to use artificial selection to advance flowering onset in target populations of the annual legume Lupinus angustifolius. Lupinus angustifolius is an annual herbaceous plant extended through all Mediterranean region, and it has been introduced as a crop in many other areas. It is a mainly autogamous plant, and its flowering onset occurs in early spring. For our study, we collected seeds from 4 populations located in Spain, 2 from a winter cold continental environment, and the other 2 from a warmer environment. And they were sown in a common garden experiment in Móstoles, in Madrid. Seeds of the 25% of individuals of each population that started flowering earlier were selected to create an early flowering line, and seeds of another 25% of each population were randomly chosen to create a control line. Additionally, we also carried out intra-population crosses between plants of the early flowering line to increase the frequency of adaptive alleles in the population and create an if1 selection line. And for all lines, we have measured the flowering onset and many other functional traits. Results show that in 2018, the progeny of the early flowering line flowered significantly earlier than the control line in all populations. And in 2019, we also found significant differences between lines for all populations. We found more pronounced differences between the early flowering line and the control line. And for both years, the advancement of flowering onset was more pronounced in northern populations. This could be due to the fact that in southern populations, flowering onset has historically been under selection for earlier flowering, and thus genetic diversity is more reduced. And the F1 selection line flowered earlier than control line in northern populations, but not in southern populations. About the functional traits, in 2018, in some populations, the progeny of the early flowering line significantly differ from the control line in other functional traits such as height. On the other hand, no significant differences were found in reproductive success. In 2019, we observed again significant differences in height between lines in some populations, reinforcing the idea that an advancement in flowering onset was related to shorter plants. And the early flowering line only generated greater reproductive success at reef population. In 2019, we also found significant differences in specific leaf area in southern populations. The early flowering line showed lower specific leaf area, which is related to more water-efficient leaves. However, the selection line showed a trend to higher specific leaf area, probably due to the presence of a greater number of heterozygotes in this line. In addition to the selection experiment, we have also performed a gene flow experiment carrying out between population manual crosses using southern populations as pollen donors. And the results also showed that for both populations, the progeny of the gene flow line flowered significantly earlier than the control line. And in parallel, we have also performed a genome capture analysis in which we have sequenced more than 1,700 sequences related to flowering process and abiotic adaptation. We wished to assess the genetic basis for phenotypic differences observed between the control treatment and the selection and gene flow lines. So finally, as conclusions, we can say, first of all, that our results support the idea that artificial selection can be helpful to advance flowering onset and create better fit populations in a context of climate change. The F1 treatment outcrossing different early flowering lines did not generate a greater advance in flowering onset. In some populations and years, the advance in flowering onset was associated to shorter plants and lower specific leaf area values. However, it only led to greater reproductive success in one population-year combination. And finally, gene flow experiment and genomic approaches will complement the assessment of the potential risks and benefits of these tools as a response to climate change. So that's all for the moment, and thank you very much for your attention.